thanks for the nice introduction, Robert, and thanks once again, everyone, for joining us here today. Our talk is about training large-scale medical foundation models, and in particular, we will focus on the pathology foundation models, as this is a current active collaboration between KAICO and the NKI.
I think we need to bring in the other presentation.
Oh, okay. I
Sorry,
thought it was already.
It should work. There. Go ahead.
Okay.
Sorry.
Cool. But let me start with the fact that oncology is really a multimodal challenge. Different data modalities are involved in getting a good understanding of the patient's disease, and in the process of diagnosis and clinical decision-making, doctors use different data types to get to the best possible treatment of the patient. So CT scans, pathology images, and oftentimes even genomics. So in this whole process, all these data types are also being generated. So the question is, how can we use all these different data types to train an AI model to solve different clinically relevant tasks in order to improve patient care in the end? Well, of course, you can imagine that if you would have a model that is able to integrate all these different data modalities, it would give the best possible representation of the patient's disease. However, a crucial step towards this first is cell membranes, and these slides can be examined under the microscope by pathologists. However, nowadays they are also digitized, making them available on the computer, resulting in these whole slide images as is shown here. And with the digitization, it made it possible to explore computational models such as machine learning and deep learning methods for a variety of pathology tasks, ranging from more supportive tasks like cell detection and classification, to tasks like tumor grading, and even more challenging tasks like survival prediction and treatment outcome prediction. However, a recurring challenge in medical AI is that we don't have a lot of task-specific labels to optimize these models. And this results in a challenge that it's very difficult to train these models in a very robust way and make them generalize well across different centers and on out-of-distribution data. On the flip side, we do have a lot of unlabeled data, especially for pathology, since H&E stains pathology slides are used in routine diagnostics of cancer patients. So, yeah, how can we utilize this unlabeled data? Well, here's where the foundation models come in, as these foundation models can be optimized in a self-supervised way, which does not require task-specific labels to optimize them, and it allows for training at a much larger scale. And these foundation models have actually already been actively researched for the pathology domain, which we show a few examples here. And as I said, they can be trained at a much larger scale and scope. And this task-agnostic way of optimization using self-supervised learning results also in a general-purpose feature extractor of which the features can be used for multiple different downstream tasks. And the promise these models have is that we actually would require less task-specific labels to fine-tune these models for downstream applications. But of course, well, compared to the natural image domain, these pathology foundation models are still in its infancies and more research is needed. Especially, you can imagine these pathology images are very different from natural images, so commonly used self-supervised learning practices may not translate one-on-one to this pathology domain. Yeah, so we need more research on that. But on top of that, these whole slide images are also huge. One single whole slide image can exceed 100,000 pixels in width and height. So you can imagine such a whole slide image does not fit into the computer memory. So a solution that is used is that we cut this whole slide image into smaller patches, so-called tiles, and these patches are usually 256 pixels in width and height. So much, much smaller, actually, than this whole slide image. And this comes with its own challenges. From the modeling side, since we lose context and spatial relationships between these tiles, but also on the more logistic side, since we need a way to acquire these tiles and store them. Doing this at scale can really matter. We have seen that in the natural language processing field, but also in computer vision on natural images. And to illustrate that, I've taken the figure here from DynoV2, where they show that by just scaling up the model size of the DynoV2 model, they can actually increase the performance of this model on downstream tasks. And also comparing different model sizes of models in the literature, we see the same trend. But with increase in model size, you also have to increase the data size. It becomes more beneficial to train on larger data sets. In the field of pathology, we have also seen that pathology foundation models, they became bigger, but also data sizes increased, as is shown in this graph here, with the largest pathology foundation model to date being trained on 1 million whole slide images.
Thank you, Vanessa. Indeed, so we see these models getting larger and larger. That is a general trend. So how do you actually train models on such a large scale? Well, there's some questions you have to look at. The first one is the training configuration. The second one is infrastructure. And the third one is how do you actually evaluate these models in an accurate and consistent way? Let's dive into these. So the first one, training configuration. We looked at, for example, the training algorithm. We compared Dino and Dino V2. And in the paper on Dino V2, it seems clear that it performs better than V1. But actually, on a limited size data set that we compared it did not show an advantage. And actually, Dino V1 worked better. This may, however, be due to the size of the data set. And for segmentation, as opposed to classification, we did see a preliminary advantage. So yeah, that's an initial finding on the algorithm. Then the model size, I'll go into that in a moment. Data selection, if you get data from a hospital, then there may be a lot of data for one disease, but a lot less data for another disease. So the data sets can be very unbalanced. So you have to do some data curation and make sure that you get a representative and diverse set of data. And also at the patch level, if you look at these small patches of data that Vanessa showed, there's different types of patches that occur in the data. And it's important, well, that distribution can also be very uneven. So there you can also look into training on a more uniform distribution over these different patch types. We actually look at a self-supervised method to do the patch type classification first as a first step, and then use that as a basis. And that seems to show some mild advantage. And we evaluated these choices of the training configuration on a relatively small dataset, TCGA. This is 10,000 whole slide images, and each of those has 10,000 patches, as Vanessa showed. So that's in total 100 million patches. So that's about 80 times ImageNet, and that's, yeah, this may sound, it's a lot of pixels, but it's just 10,000 patients. So yeah, it's still, in terms of number of patients and the diversity that you see, relatively limited. Now, we compared a lot of different options and evaluated them, and some of the things we found are what you would expect. For example, we looked at the dataset size. If you take different subsets of TCGA, then yeah, the larger the subset, the better the performance. That's what you would expect. But there were also a few things that that were not immediately obvious up front. So let's go through a few of those. The first one, we looked at resolution. So these pieces of tissue, they're put under a microscope. And if you think of 1 millimeter of tissue, for example, 1 millimeter of your skin, then there's 4,000 pixels in there at 40 times magnification under the microscope. So that's, yeah, 4,000 times 4,000 is 16 megapixels for a square millimeter of tissue. So it's really, yeah, one high resolution picture for that tiny piece of tissue. So very high resolution images. That also means that you have a very small area that you're actually visualizing. And that may explain why for the 4 tasks that we compared here, so Bach and Pascal-Median are in breast cancer, and MHIST and CRC are colorectal cancer. This very high level accuracy, resolution, in blue here, gives the lowest performance. If you zoom out by a factor 2, so 2,000 pixels per millimeter, in yellow, then you see that has intermediate performance. But what we found gives the best performance is to actually train on also low resolutions and higher resolutions and mix them all together. So 5, 10x, 20x, and 40x. So that was the first interesting finding that we got. Then what do these models actually learn? And yeah, this I personally found very insightful. So we ran inference with the trained foundation model on the whole data set. And then you get a set of embeddings, that's just a bunch of vectors. And we project these to a 2D space using the t-SNE method. And then each embedding is just a point in this 2D space. And then you can color these points with different labels that you may have. So here, for example, for TCGA, we know the disease. And we can see that at the top right here in the left finger, all the orange pixels, the orange patches, they are all from the same disease, which is glioblastoma. So you see a very clear clustering here in the left image, which means that the model has learned to organize its embeddings in such a way that they represent the cancer type. So that's a very good thing. It clearly learned a biologically relevant feature. Now, you can do the same exercise, but now instead of coloring with disease, with cancer type, you can take the hospital where the data came from. And to my surprise, you then also get a very clear clustering. I think that's not necessarily desirable, because when you look at that same glioblastoma, you may see here that there's two parts. And this actually explains why there's two parts. So the top part is from Duke University, and the bottom part, which is blue here, light blue, cyan, is from Henry Ford Hospital. So the model has not only learned to tease apart different cancer types, but also different hospitals. And there's actually a lot of things you can learn from this image if you look more closely. For example, in cyan you see uterine cancer. It's in 3 different places. Well, you'd hope these would be in one location together, right? But here you see why that one is up there and not closer to the middle one. And that's because it's close to the other orange blob there. It's one big orange blob because it's from the same hospital. So there the model chose to group these data points from the same hospital together instead of data points from the same disease. Now, we also looked at model size, so just to validate that the models we train have good performance compared to the current state of the art. The top 2 lines are existing models from others. The first one by Kang from last year. The second one is Vicon from Okin. And these are models similar in size, similar data set size. And the next 3 models in blue, those are just a bit small, a bit base, and a bit large that we trained. And in blue you see the best performance for 5 different tasks. And you see that actually for each of the 5 tasks our models got higher performance than the comparison models. So that shows that they perform at the state of the art, or slightly above it. And it's also interesting to see that the fit base, that size, has the best performance for 3 of the 5 tasks. So that seems to be kind of the right fit for this size of data set. So fit large is probably too large for this relatively small data set. But when you start to train on bigger data sets, then probably fit large will outperform fit base. At the bottom you see 2 larger models that perform better, but those were trained on much larger data sets. For instance, Virtuo is the one that Vanessa showed at the top right with 1 million whole slide images. And those models are not public, so we could not evaluate them ourselves. That's why they're in gray, because they're not directly comparable. Second topic, infrastructure. So what many people did typically so far in machine learning and in kind of classical machine learning is to always start with generating patches. And that's a time-consuming step. I've been in projects where only that step alone took a whole week on the whole cluster. Also, moreover, it limits the patches to a fixed set. So typically you choose a grid, and then that defines which patches you get. But each of these patches, you could just as well have shifted it a little bit to the left or up or down, and you would have a different patch, but that will not be represented in your set if you choose a fixed patch set. Moreover, it also duplicates your storage requirements because you have to store this whole set of patches. So to avoid these limitations, we developed an in-house procedure for online patching so that we have a more flexible setup. And here we randomly choose a patch from anywhere in the whole slide image. So that increases the diversity of your data set. It effectively increases the data set size because now any patch is possible. So that's actually 16,000 times magnification of the number of possible patches. Of course, many of them are very similar, so that number should be treated with care, but a lot bigger at least. And also, it makes it a lot easier to do experiments like the one I showed with multiple resolutions, because you can just generate those on the fly. It's going to be much too expensive if you have to generate all those patches up front. So we developed a method to stream this data efficiently from cloud storage, and that will be very beneficial if you scale up the trainings. Now, why would you scale further? Well, let's think back about the goal that we were describing here. We want to improve the life of patients, so you need models that perform as well as pathologists or better. For this, we clearly need to scale because the current pathology models are not there yet. Also, cancer has many different subtypes. In fact, even just within lung cancer, there are some particular variants of lung cancer with a particular genomic signature where only 1% of the patients have it, but you can perfectly identify those patients by the genomic signature and have special medicines that work for that particular group of patients. So you can imagine if you need a representative data set for each of those different patients groups, for all the different cancer types, you need a lot of different patients. Also, the features that you're looking at in the images, they're small, so only a small part of the image may be relevant. So where are we now? What we showed was results on TCGA, which is 10,000 or 30,000 slides, depending on which variant you choose. We're currently at a stage where, together with NKI, we're training on 100,000 slides. We're scaling up to 1 million, and then together with NKI, we also want to scale it up further with multiple institutes. So we're actively looking for cooperations with other hospitals to grow this data set into a multi-center data set of going up to 10 million slides. When you scale up, there's some questions that you need to explore. So I mentioned data curation. You have to find a diverse and representative set of the data. Also data sampling, like the patch sampling that I discussed. Will the robustness issues that we saw, where the hospital plays such a big role in what the model learns, will that go away if you increase the scale? That will be an interesting question to see. And in any downstream task that you train, you have to evaluate whether there is a bias, or whether, for example, the medical center does influence this. And just to give an idea why these differences occur, the staining of these images, it varies per lab that does the staining, so the coloring of the image, and that can explain why you so clearly see the hospital reflected in the embedding space. We're expanding our cooperations, so we're actually diversifying the data. At the moment, we're training a radiology foundation model. We're also doing experiments on RNA and DNA and getting into vision language models. We also think that an important question will be how can you let these models reason in a more reliable way? Because current LLMs clearly are not yet able to do that, reason in a reliable way. So we believe that's a very promising and important research area. And we're partnering with other organizations to train and validate. So we're also starting now with 2 projects where we already bring this into the clinic. So the first one is about cell segmentation. So you put in an image and then the model will segment it for you, segment the different cells, and then color them to do a classification. And the second one, copathy, is a pathology assistant based on AI, where the pathologist can talk with the AI model, and also this can produce structured reports. We are committed to contributing to open science. So the results that we showed, they're being made available in a paper. We just submitted this to arXiv yesterday evening. It should appear there tomorrow probably. The model is being released, and also the evaluation framework that we used to generate these results. And in the future, we'll also make available data and annotations. So I already spoke briefly about evaluation. Well, evaluating foundation models is hard. Why is that the case? Well, you cannot evaluate these embeddings directly. You have to train on a downstream task, and you also need to evaluate on multiple downstream tasks, because you want to see, is the model able to do well on multiple different tasks that are clinically relevant. Another issue is that if you let 2 different teams evaluate one foundation model, they will almost certainly get different results, because there's small details in how you set up, for example, the linear regression or the downstream model. And so it's really important that different models that are being trained by different groups, that we can see which model performs better than which other, so to evaluate them in exactly the same way. That's why we believe you need absolutely an evaluation framework that is used by multiple teams around the world, or at least to evaluate all the models produced by those teams. So that is why we're open sourcing our framework. It's called EVA, or EVA, I don't know, Roman, how you pronounce it. But, all right. And what it does for you is it downloads the data, it runs inference so you get the embeddings, and it then trains the downstream model on various tasks. And so far there's only a few. There's the 4 that I mentioned, so 2 breast cancer tasks, 2 colorectal, both binary and multi-class, Bach, CRC, MS, and Pashkinidian. But the idea is really that this will be growing because we want to cover the whole space of clinically relevant models for pathology. So cooperations are very welcome here. If you have any data set or you know of a data set in pathology that you think is useful to add here, please let us know. We're happy to add it. And also in radiology. So there's also a first data set already there. Then the good thing about EVA is it's actually already available. I'll show this QR code in a moment again. So, or feel free to take a picture now if you want. Now, we can also ask this question, what will happen when you start to do these trainings on a larger scale? We saw these robustness issues where the center is clearly seen from the data in the hospital. Will that disappear when you train on a larger scale? Maybe, maybe not. Curious to hear what you think. Another one is performance, of course. So we believe that with increasing the scale, we may at some point get to above human level. So for natural, that may sound very bold, but actually for natural images we already saw that, right? AI models can better discover or detect faces and classify them than humans can from pictures. Then in radiology, as Robert mentioned, I worked at ScreenPoint before, and then we trained a breast cancer AI model on radiology images. And there, after improving the machine learning methods and scaling up the data to 1 million patients, we found actually that these models started to perform better than the best radiologists. So these models are being used, they're FDA approved and used in hospitals all over the world. And in the Danish population screening last year, it was seen that the time it takes for radiologists when they use AI is reduced by 50%, or 44 I think, but somewhere around there. So it's a lot faster, saves a lot of time, so radiologists can evaluate more mammograms, so that's important because in many countries there's a shortage of radiologists. But moreover, 20% more cancers were found. So that's very important. Think about if you're a woman and you have breast cancer but you don't know it, if it's found earlier, you have a better chance of survival. So this really has a big impact on patient survival. So for radiology, clearly that has happened, and I think it's very clear. I expect the same thing will happen for pathology. Just not if you use only 10,000 patients like are in TCGA, but when you do scale up to 1 million patients, like it's already happening in the virtual model, we think this potential is now very soon going to be realized. Very curious to hear also about this question, what you think, but then the most interesting thing that I think we can learn from these models, they can actually teach us. So there's some subtle patterns like microsatellite instability, and then some pathologists can see it in the image, but others cannot. I think that is very intriguing. These are really experts, right? They've trained for 10 years at least and looking at these images, and as a normal person, there's no way you can see the same things that they can. But still, there's some things they can see and others they cannot, but they are present in the image. We know this because the models can see them. So there's other patterns that, for example, molecular biomarkers, so the genomic footprint or RNA footprint, that these models also can see. So apparently the information is in the image. So if we study what these models get from the image, then we can maybe learn new biological insights. I think it will be very intriguing, and curious to hear what you think. Thank you. So curious to hear your thoughts about that, and also if you have questions, let us know.
Hi, thank you. Oh. On slide 13.
Let's see. 13. Wait, 13?
13.
Yeah, okay.
So the presented results seem very close to each other, where I was wondering about confidence intervals here. Yeah. More generally, obviously the question, with language models, the great adoption was there is very low risk in being wrong. Here, obviously, there's giant risk. Yeah. So how do you guys deal with confidence intervals or causal inference, et cetera?
Yes, very good question. So there's different types of confidence intervals that you can measure. So when the AVA framework does a linear regression, there's a random seed, and when you run it multiple times, you get different results. We compute a standard deviation, and that is reported. So that's already a first way that we evaluate the standard deviation. But then still, if you take a different train-val split of the data sets, you get a different result, right? But for many of these tasks, like patch chameleon, the split is fixed. So if you want to compare with others, you have to report on exactly the same split. So that limits what standard deviations you can calculate there. And then there's yet another standard deviation, and that's also a very important one. If you repeat the foundation model training 5 times or 10 times, what distribution do you get? Well, that's a very expensive one to estimate, but it is an important one, so it's a good question.
Thank you so much.
Sure.
I have one question about the batch effects that you showed
Yes.
with the TSNE.
Yeah.
So we know that you have medical centers that are specialized in given indication, so you don't know whether the real signal is the medical center or the indication you're looking at. So I was
Sorry, I did not understand that. They specialize in what?
Sorry, you have some of the medical centers in the US mostly are specialized on given indications.
Ah, right, yes.
So what you show here on the right-hand side is that you might be getting some information about, for example, the microscopes that have been used in the medical centers rather than the biological signals themselves.
For example.
So I was wondering whether instead of making these plots and analyzing these on the images themselves as tiles or the host-like image, you were trying to predict things like what is tumor and what is adjacent normal tissue that you normally have in the tissue to be able to actually validate that you capture real biological signals rather than more of these batch effects.
Very good question. I think that's a good one to also measure. And indeed, already this plot also shows what you mean, I think, because in dark blue at the bottom left, you see that sarcoma, there's only one blob there, but there's also only one hospital. So it's only one hospital that does sarcoma. That's what you mean, right?
Mm-hmm, yeah.
So there is some correspondence there. But indeed, I think it's very useful to do similar coloring for different biological features that you know. So any labeling that you have available in the metadata is useful to do similar exercise.
And are you working like with pathologists to also validate those type of findings, like having the overlays of the images and then showing to a pathologist and say, does my model capture the right region for?
Absolutely. So that's really crucial to the whole way that Kaiko is set up. We work very closely together with the NKI and also in future with other multiple hospitals. And we have close contact with pathologists and with the research groups of NKI. And together always evaluate these results because just as an AI company you would not be able to do clinically relevant things. So that's essential. Yeah.
Thank you.
I don't know if there's time. How are we doing on time, Lydia? Right. Jonas?
Yeah, so how do we deal with the new targets? I mean, with this model you could try to predict how a patient would respond to a certain treatment, right? So that's an important question. But these treatments keep on changing over time. So how would you deal with the foundation model for this problem?
Yeah, that's tricky. Then you already at least need data about what treatments the patient's got. You need to segment the patient population, and that will reduce your data set. So that's a hard problem. I don't have a finished solution for you.
So I mean, you could probably get the data where you have like 50 samples or something like that.
Then there's not so much you can do, I think. Unless, there's ideas to think about.
But there's also more on the evaluation side of it, right? Because the pre-training can happen still in all of this data. But of course, the evaluation becomes more features.
Good. Thanks again.